Sir, we are live now. Shall I start?
Yeah.
So good evening, doctors. Shumon on behalf of Shield Healthcare, welcoming you all in today's a very interesting topic on male factor infertility, and our guest speaker is Dr. Badrul Haque, sir. It is my honor to introduce sir. Sir is the obstetrician and gynecologist and senior consultant in Milton Polyclinic and Diagnostic Center, Hojai, Assam. With the short introduction, I request you, sir, to take over the session. And meanwhile, I request all the participants for your active participation, and please post your queries on the QA dashboard of the Shield Connect page. So once the talk will be over, we shall have a short discussion on this topic with sir. So thank you very much, sir, and over to you, sir.
Yeah. Good afternoon, everyone. Today I'm going to present a webinar on male infertility, male factor infertility. So I hope I can justify on this slide. Okay. So male factor infertility. Male factor infertility, its incidence is, it is in that male factor is responsible for 40 to 50% of all cases of infertility, and it is increasing at an alarming rate. So primary pathogenesis of male reproductive system involved in male infertility is that male accessory gland infection, prolonged stasis of spermatozoa in epididymis or in transit, immature public abnormal spermatozoa. So there are environmental and lifestyle factors like drugs, smoking, pollution and radiation, and systemic pathologies like diabetes, cancer, and systemic infection. All it is seen that all this accumulates and leads to oxidative stress, and which leads to spermatozoal dysfunction and hence infertility. So we have to evaluate for oxidative stress and treatment of underlying pathology, antioxidant supplement, and avoiding factors promoting ROS in value, reactive oxygen invalidities. So causes of male infertility. It is seen that idiopathic accounts for 31.7%, varicocele 16.6%, infection 9%, hypogonadism 8.9%, cryptorchidism 8.5%, malformation 5.8%, systemic 5%, immunological factor accounts for 4.2%, tumor 2.3%, obstruction 1.5%, and others 6.5%. So management. There are two modalities: surgical and medical management. In surgical, we have to repair the varicocele, or if there is any tubal block in the pathway where sperm travels. So if there is any tubal block, we go for surgical repair, and if there is varicocele, we go for its repair. And medical treatment, we treat the hormonal imbalance and erectile dysfunction later. And androgens, HCG, HMG, FSH, systemic corticosteroid, homocaptain aromatase inhibitor, and antioxidants. Role of oxidative stress in male infertility and updated review. Current evidences links oxidative stress (OS) to male infertility. Okay. Current evidences links oxidative stress to male infertility, reduces sperm motility and sperm DNA damage, and increased risk of recurrent abortion and genetic death. Oxidative stress is detrimental to fertility, pregnancy, and genetic status of the newborn. Oxidative stress and testicular microenvironment. It was found that oxidative stress affects negatively the testicular microenvironment and ends up causing a deficient spermatogenesis, damages to sperm DNA, as well as reduced motility and altered morphology of spermatozoa. Idiopathic male infertility is characterized by increased reproduction of reactive oxygen species, which causes the depletion of sperm antioxidant. Physiologically, spermatozoa have a low antioxidant capacity, and for this reason, they can be easily affected by ROS, that is, reactive oxygen species. Antioxidants for protection. To help sperm function, particularly in case of idiopathic infertility, a constant supplementation with an antioxidant starting from sperm cell development to conception is fundamental. These molecules are free radical scavengers and protect spermatozoa by counteracting lipid membrane peroxidation, protein corruption, and DNA fragmentation. There is original article. Systemic review of antioxidant type and dose in male infertility. Benefits on 7 parameters: advanced sperm function, assisted reproduction, and live birth rate. Now, result: in all, 26 studies reported a significant positive effect of antioxidant therapy on basis of 7 parameters: advanced sperm function, outcome of assisted reproductive therapy, and live birth rate. Antioxidants, mechanism of action, and rationale for use. A large number of compounds with antioxidant properties have been investigated for the treatment of infertile men. An understanding of antioxidant mechanism of action of commonly used compound is important before embarking on the evidence associated with their use in clinical practice. First compound, L-carnitine. L-carnitine is involved in sperm metabolism, fueling important activities like sperm motility. The main function of L-carnitine in the epididymis is to provide an energetic substrate for spermatozoa, may be involved in successful maturation of sperm. L-carnitine is necessary for transport of fatty acid into the mitochondria to produce energy. Low level of L-carnitine reduces fatty acid concentration within the mitochondria, leading to decreased sperm motility. Water-soluble antioxidants involved in sperm metabolism, fueling important activities like sperm motility. Indeed, in vitro studies of sperm culture in media containing carnitines had higher motility and viability in comparison with control. They exhibit their antioxidant activities through scavenging superoxide anion and hydrogen peroxide radicals, thereby inhibiting lipid peroxidation. Significantly lower carnitine level were observed in semen samples from infertile men with oligoasthenoteratozoospermia. Coenzyme Q10. It is vital antioxidant omnipresent in almost all body tissue. It is particularly present at high concentration in sperm mitochondria involved in cellular respiration and plays an integral role in energy production. This contribution rationalizes its use as a pro-motility and antioxidant molecule. Furthermore, it inhibits superoxide formation, delivering protection against OS-induced sperm dysfunction. OS-induced. A significant negative correlation between coenzyme Q10 level and hydrogen peroxide has been reported, and a linear correlation between coenzyme Q10 level in plasma sperm counts and seminal plasma sperm count and motility was detected. Now it's selenium. Selenium is an essential trace element whose involvement in spermatogenesis is thought to stem from its ability to protect sperm DNA against OS damage. Selenium is a major constituent of a specific group of protein called selenoenzyme. Its antioxidant properties are thought to stem stem from its ability to augment the function of glutathione. More than 25 selenoproteins exist, such as phospholipid hydroperoxide glutathione peroxidase and sperm capsular selenoprotein glutathione peroxidase. This helps maintain sperm structural integrity. Selenium deficiency has been most commonly associated with morphological sperm midpiece abnormalities and impairment of sperm motility. Zinc. Zinc is another essential trace element with biological role. It plays a vital role in metabolism of RNA and DNA, signal transduction, gene expression, and regulation of apoptosis. Its antioxidant properties are thought to result from its ability to decrease production of hydrogen peroxide and hydroxyl radical through antioxidant redox active transition metals such as iron and copper. Zinc concentration of seminal plasma were found to be significantly higher in fertile men in comparison with subfertile men. Zinc is thought to deliver an important protective effect on sperm structure. Sperm flagellar abnormalities such as hypertrophy and hyperplasia of fibrous sheath, axonal disruption, defect of inner microtubular dynein arms, and abnormal or absent midpiece have all been associated with zinc deficiency. Now it's lycopene. Lycopene is a bright red pigment and phytochemical found in tomatoes and other red fruits, watermelon, guava. It belong to a class referred to as carotenoid, which are yellow, orange, and red pigments synthesized by plants. Lycopene, a naturally synthesized carotenoid present in fruits and vegetables. It is powerful ROS, quenching ability make it major contribution to human redox defense system. Lycopene is detected at higher concentration in human testes and seminal plasma with levels that tends to be lower in infertile men. Sperm treatment with extracellular ATP increases fertilization rate in vitro fertilization for male factor infertility. In conclusion, the present study shows that extracellular ATP can induce an increase in human sperm fertilizing potential and provides a biological rationale for use of these nucleotides for the in vitro treatment of human spermatozoa before oocyte insemination during IVF techniques. Further studies with a larger number of couples will be necessary to confirm the beneficial effect of this procedure. Treatment of oligospermia with amino acid arginine. This slide shows the parameters. Severe oligospermia, less than 20 million per ml. Before treatment, it was 66. After treatment, it is 22. So this is a study. Result of treatment of arginine shown here. Before treatment and after treatment. Now unveiling the unknown. Fact 5. Freshly ejaculated spermatozoa are incapable of oocyte. The fertilization aptness of spermatozoa depends on the appropriate and time-dependent acquisition of 3 important biological processes. These 3 biological processes are capacitation and hypermotility, chemotaxis, and acrosome reaction. Capacitation. Capacitation is a functional maturation of the sperm. This process starts from uterine activity. The sperm head is surrounded by glycoprotein. In capacitation, glycoprotein are removed makes sperm more active and gain hypermotility. Capacitation reduces binding affinity of sperm in the female reproductive tract. Chemotaxis, the travel guide. Chemotaxis plays an important role for fertilization. Chemotaxis refers to direct movement of the sperm to the oocyte. Oocyte releases chemoattractants. With calcium ion oxidation, sperm is directed towards ovum by chemoattractants. These are the two diagrammatic representation. Acrosome reaction. For fertilization, the sperm has to penetrate the following oocyte layers: cumulus ophorus, corona radiata, and zona pellucida. After transit through the corona radiata, after transit through the corona radiata is completed, the sperm binds to the zona pellucida at Z3, at zone 3. Acrosome reaction. After binding, the sperm and ovum plasma membrane fuses. Spilling of genetic material from sperm to oocyte occurs. Fertilization follows. Hence, fertilization follows. Three biological process depends on calcium ion oscillation. The fertilization, three biological process depends on calcium ion oscillation. The fertilization aptness of spermatozoa depends on appropriate and time-dependent acquisition, chemotaxis, capacitation, and acrosome reaction, where calcium is extensively involved in almost every step. This is the diagram flowchart showing spermatogenesis. These three biological procedures like capacitation, chemotaxis, and acrosome reaction, where calcium ion oxidation is almost required in every step, and it depends on myoinositol. Myoinositol, a new hope, a new impetus. Myoinositol vital for fertility. The concentration of myoinositol in seminiferous tubule fluid is higher than the level found in seminal plasma. Myoinositol in mitochondrial membrane potential. Myoinositol in mitochondrial membrane potential. Myoinositol also directly increases the mitochondrial membrane potential, that is an apoptotic marker in the sperm cell. Myoinositol seems to have a beneficial effect on motility, fertilization capacity, and embryo quality, as demonstrated by in vitro studies on sperm of normal and OAT patient undergoing IVF. Myoinositol decreases DNA fragmentation. The impact of in vitro semen supplementation with myoinositol upon DNA fragmentation was resulted in a cross-sectional study done in men and presented at 2015 ESHRE meeting. Collaborates, actually this slide is not coming clear. Although a preliminary report, a significant overall decrease in DNA fragmentation was determined in myoinositol treated sperm, although not expanded to larger group. Slide is not very clear. Clinical study. Myoinositol on idiopathic infertility. Myoinositol improves sperm parameters and serum reproductive hormones in patients with idiopathic infertility. A prospective double-blind randomized placebo-controlled study. Now the study comes. Clinical study. Method. Double-blind randomized placebo-controlled trial. Place Italy. Number of patients was 194. Myoinositol 98 and placebo 96. Doses given as myoinositol 4 gram per day. Duration was for 3 months. And publication was American Society of Andrology, February 2015. It aims to find out the efficiency and safety of myoinositol treatment in men with idiopathic infertility. Myoinositol significantly increased, the study reveals, myoinositol significantly increased percentage of acrosome reacted spermatozoa, total count, sperm concentration, progressive motility, myoinositol rebalances, serum luteinizing hormone, follicle stimulating hormone, inhibin B concentration. Thank you.
Thank you very much, sir, for the presentation. Just give me 1 minute time to check if any question is there in our dashboard, sir.
Hello.
Sir, I can see one question. Sir, am I audible? Sir, am I audible?
Yeah, yeah.
Yes, Suman.
Breaking a bit.
Yeah. Just. So, sir, the first question is, sir, what is your practical observation with this myoinositol in the treatment, means male infertility treatment, sir?
So far, we have found out good results with myoinositol treatment in male infertility.
Okay, sir. So, there, sir, is there any specification or is there any particular clinical aspects where you can give this myoinositol or something like that? Or generally, in terms of this, if the semen parameters are not well, then only you can give this myoinositol for improving the sperm parameters.
Actually, the voice is breaking. Repeat again.
Sorry. Yes, sir. Sir, I'm asking that.
Yes.
Is there any special consideration or is there any special scenario where you can give this myoinositol, or in terms of the general sperm parameters?
In terms of general factors for male infertility, you can go for myoinositol.
Okay, sir.
We have found some good results previously
with myoinositol, sir. And
yeah,
sir, may I know, sir, this how long can it be prescribed to treat this male infertility? What is the optimum time period?
Duration we have given for 3 months and extended up to 6 months.
That means up to 6 months. What is your feedback? Up to 6 months is okay.
We got some
for improving the sperm parameters and
if sperm count increased and all other morphological defects.
Yes, sir. So, does it have any effect in this case of DNA means fragmentation index?
No, this I can't say at this moment. I don't have any idea about this. Hello.
Okay, sir. Are you getting find any other question? Yes, sir.
Your voice is breaking, and I'm not getting it very clearly.
Yes, sir. We are not. I cannot see any other question in our dashboard.
Okay.
So, if you allow me, then we can conclude this session, sir.
Yeah. You can. I also have some.
Thank you very much, sir, for giving some time in this Shield Connect platform and for such a nice presentation on this male infertility. And in future also, we will look forward for some valuable session with you, sir, on our Shield Connect platform. So thank you very much, sir, and have a good day, sir.
Thank you, sir.
Thank you.
Thank you, sir.